So a common setup of AI models is to take a large array of predictors, here genomic data, omic data, maybe blood markers, imaging, and even real-time data, to make a decision, either prognosis or diagnosis or treatment response. And there are two reasons to do this. One is if you actually want to make the decision, for example, to identify fast tau progressors, people who have tau accumulation in the brain, to enroll them in a drug trial. Or you might want to discover new predictors in the genome or in other biomarker data sets that can help you in making these decisions. And if they increase risk, they can become treatment targets. Now, deep learning methods are a type of AI method. They include convolutional neural networks, generative adversarial networks, transformers, and latent diffusion models. And these can be specially adapted for handling images or genomic sequences. Let's say we wanted to build a computer vision system that recognized a person. This is a person called Sarah. And if we had a lot of images of her and images of other people, we could train a convolutional neural net to identify features that are aggregated across layers of the neural network to discriminate between her and other people. And when we adapt this to 3D medical imaging, the decision is not who it is, but it's the person's diagnosis, whether they're amyloid positive, whether they have Alzheimer's disease or vascular pathology or something else. Now, this truly remarkable paper about a year ago, we published with Bin Liu and Xiaogan Yan, that centralized Alzheimer's disease and control MRI data from 217 sites. This is one of the largest MRI studies ever performed. And this was able to classify new scans that it hadn't seen as being from patients with Alzheimer's or healthy individuals with over 90% accuracy. And this is tremendously good news. So we've been benchmarking deep learning methods to detect Alzheimer's in images, really comparing the accuracy of different approaches. This is one that derives so-called radiomic features, texture, various features from the histograms of the images. We've been trying deep learning methods based on sequence artificial intelligence, so-called LSTMs, that read in a series of image slices and make a decision. And also we've been adapting to 3D popular computer vision models such as DenseNet, which are often pre-trained on photographs, MRIs, and with various sophisticated methods. So in this work led by Nikhil Dinagar in our group, we found that obviously as you get more training data, the ability to detect Alzheimer's disease in a scan goes up. But some of the methods really outperform many of the others. One of these methods with a 3D convolutional neural network gives 91% performance. And there's been huge improvements in detecting Alzheimer's disease, even with a fairly limited amount of training data. More recently, we've been trying so-called vision transformers. A transformer is the deep learning module that drives large language models such as ChatGPT, but we've been handling it, adapting it to handle 2D or 3D MRI scans. This new paper from our group, Nikhil Dinagar as first author, shows that these so-called vision transformers can actually give very, very high accuracy in detecting Alzheimer's disease in a scan. And they're trained, I mean, just out of interest, on synthetic MRIs as well as MRIs from patients. This is a latent diffusion model that can generate MRIs, and it's been found very beneficial. Okay, so these all are a little bit of a black box. You can't tell how they're working. So to make them more interpretable, Li Shen's group and Dok-Yoon Kim's group at University of Pennsylvania have been developing deep learning methods that take SNPs or genetic variants, relate them to variations in brain volume, and then onwardly relate those variations in brain volume to the AD risk score for a particular individual. Now, these are a little bit more interpretable, and they give very good performance, not just in predicting Alzheimer's disease, but informing us about the regions of the brain and the genome that are relevant. Can you predict who will get Alzheimer's disease? So this remarkable work combines neuroimaging and genome-wide SNP data, and tells whether you will be developing Alzheimer's disease or converting from mild cognitive impairment to Alzheimer's disease in the next 2 years. This outperforms many related machine learning methods by a significant margin. It copes with missing data, and it even tells you what type of cognitive deficit the person has, whether it be in memory or language or executive function or visuospatial aspects. This work by Dugu Tosun at UCSF uses baseline data to predict who will accumulate tau, one of the major pathologies of Alzheimer's disease, in the future. And so she's been training machine learning models to decide whether or not a person will accumulate tau pathology quickly, moderately, or not very fast at all, to identify fast progressors. Now, she found that moderate accumulators of tau and fast accumulators of tau differ in that they have a greater baseline presence of white matter lesions, psychiatric symptoms, even a greater amount of amyloid and tau in the brain to begin with. And you could imagine that this could lead to more efficient drug trials. If you were guided by this information, you could enroll people that would decline more quickly. And obviously, this can lead to much more efficient drug trial design to detect the same effect. Related work, can structural MRI be used to detect amyloid in the brain? This work by Tamong and Chattopadhyay has been predicting amyloid positivity from cheaper and more widely available MRI scans. Diffusion MRI performs particularly well. And he's been using a so-called Y-shaped hybrid neural network to also include demographic and genetic data as predictors. This work by Yan Jin, in collaboration with us at Biogen, has been had a patent filed on it by Biogen. It can create a synthetic PET scan, an amyloid PET scan from multimodal MRI. MRI is a lot cheaper and much more widely available and less invasive. And imagine if you could create PET scans for people with just an MRI. You could allow massive screening of huge databases of MRI. And this actually seems to be quite promising in reproducing on data that's been held out the profile of amyloid in the brain when you have ground truth available to verify it. This principle of computing a scan that you didn't collect is often known as neural style transfer. You can use it to enhance imaging or make a scan look like it was collected on a different scanner. And one very powerful application is for harmonizing MRI. This work by Nangting Liu and Neda Jahanshah just out converts MRI data to appear as though it was collected on a different scanner. It's a deep learning generative adversarial network. And one of the benefits of this is that the classification accuracy of Alzheimer's can be improved if you feed in images that have been harmonized to match the training data. Now, how is all of this neuroimaging AI relevant for genetic target discovery and drug discovery in Alzheimer's disease? Well, this work by Taiho Zhou and Andy Sakin in Indiana asked the following question. It said, where is it in a tau PET scan that the accumulation of tau is particularly pertinent to deciding whether someone has Alzheimer's disease? Now, these signatures that are learned by a neural network can then be ranked in future people and genetically screened to find genomic markers that promote the accumulation of tau. One such approach, the sliding window association test, uses a CNN, that same convolutional neural network that we saw applied so successfully to images. And Taiho Zhou's method slides along the genome and finds particularly predictive signatures in the genome for diagnosing Alzheimer's disease. And compared to standard univariate methods such as PLINK, it's very effective in finding novel signatures that hadn't been anticipated before. This can also be extended using a transformer module. Attention-based deep learning can detect genetic motifs linked to Alzheimer's. The difference here is that attention allows the aggregation of information across the genome when you're trying to make a prediction of Alzheimer's disease. So we'll be benchmarking these and other genomic discovery methods as part of our one-year supplement to work with the other AI and machine learning awardees. And one of the nice things is this provides interpretable maps of the genome in terms of which features it's been seeing and aggregating to make a decision. To summarize, we made a tutorial for the ADSP, it's on YouTube, on genetic analysis of biomarkers, imaging being one such biomarker, but it also talks about tau and amyloid in the CSF and plasma. We also led genome-wide association studies that discovered in the last year loci in the genome associated with faster brain tissue loss. So this particular paper in Nature Neuroscience last year finds 15 markers in the genome, common variants that are associated with the speed of brain aging. Of course, these are very attractive drug targets for the future. Another paper under review at Nature Genetics, which we co-led with Enigma and the Charge Consortium, discovered 51 genomic loci that are associated with hippocampal volume, a very well-known biomarker of AD. And we're now looking up the overlap with the loci in the famous AD GWAS by Brian Kunkel to see which markers exert their effect on AD through promoting hippocampal atrophy. ApoE4 was discovered to shift hippocampal volume negatively in an age-dependent way. And Christos Davatzikos and Zhenhao Wen at University of Pennsylvania also discovered 4 subtypes of atrophy in the brain that are associated with different and distinct clinical CSF and plasma signatures. And this is exciting for genetics, because if you could analyze the contribution of different processes to aging and dementia, you could essentially give each one a score and do genomic analysis of those scores. And so they've been doing genome-wide screening of the coefficients or the loadings on these profiles estimated from new individual scans. They also suggest that there's an early profile of atrophy, very diffuse, in the brain that is promoted by genomic markers related to hormonal, cardiovascular, and immune processes, and also a subsequent progression that is more focused on the medial temporal lobe, that is more promoted by genes that influence tau and amyloid deposition in the brain. They've been working on a newer method that clusters both the images and genomic data simultaneously to find joint sets of biomarkers in the image and the genome that influence AD. And they've been adding these to predictive models, obviously using the imaging subphenotypes, and all of the available plasma and genomic markers helps you to predict who will decline, as is valuable for clinical trial stratification. So last of all, can any of this be used to find drug treatments that counteract the decline? So Gyunga Chun has been working with Christos Davatzikos to identify novel polygenic risk scores that are associated with faster decline. And she has found that if you look for genomic markers that are particularly expressed in certain types of cells, in astrocytes or oligodendrocytes, they have particular patterns of action in the brain. This is work with Priya Rajagopalan at USC, where she's mapped a microglial polygenic risk score into the brain and associated a certain pattern of atrophy with it. And here's another one where a certain pattern of atrophy is linked with a different module. Then Gyunga Chun has been working with Julia TCW in the Functional Genomics Consortium to literally apply repurposed drugs that counteract the implicated pathways in these genetic modules. And this is very important and novel work that gives a novel translational aspect to this gene discovery work. Last of all, the tools I discussed have been presented at over 20 symposia, workshops, and panels in the last year, notably the ones that are listed here. And some final questions we'd like to ask: are there any additional AI questions that we should be focusing on, such as predicting anti-amyloid treatment response, or so-called ARIA, a treatment side effect of anti-amyloid drugs? And secondly, are there any public data sets that can be used to answer these questions? Thank you for your time. We really appreciate your support.